Dear viewers, Assalamu alaikum. In our previous video, we studied that how we can use different online servers for the prediction of function of a protein. In that video, we discussed a cofactor, which is an online web server which can be used for the prediction of a protein, and also iTasir or some other tools. So here we will discuss the results that we obtained from the tool which we used last time, that was Cofactor, and from that server we will get our results after some time. So here we obtained our results that we submitted. So we will discuss all these different options that we can obtain from these results one by one. Let's suppose we have submitted the 427 amino acid residue sequence, which is shown here, and we can again download this query sequence and the structure, and we can obtain or download all the results from this tool. So let's see that what we will obtain. So protein with similar structures. So here we have 10 different proteins with PDB ID and the alignments which we can obtain according to our query structure. Let's suppose we want to see that what is that protein which is closely aligned with our protein with RMSD value of 0.59 and TM score of 0.92, and all these structures are given here with line models, and the ribbon model is our query structure. We can click this to view our protein structure, and we can directly open it in PDB file, and also we can download the structure, like here it is downloaded, and we can see that what is that protein in Discovery Studio visualizer. And this is our protein, which is the three-dimensional structure of porcine pancreatic procarboxypeptidase. So this our protein mostly closely aligned with that protein which is given here, and also it gives 10 other proteins which can be aligned or which are closely aligned with our protein, which are the analogs in PDB. So these are the top 10 structural analogs in PDB. Now it also gives us predicted gene ontology terms, in which it gives us molecular function through gene ontology. So it gives two different functions for our protein, which is our query protein. One is metallocarboxypeptidase activity, and the other is zinc ion binding activity. So these two activities have been shown by this functional protein, the protein for which we predicted the function. We can also download full results from here by clicking here, and we can also get this image. When we click on this image, it will give us the full and high resolution graph. Similarly, biological processes, this protein is involved in different biological processes which are shown here with different C-score. So C-score is through gene ontology tool, and it gives 0.944 for proteolysis. So the major function of this protein is proteolysis, protein breakdown, and then 0.78 with cellular processes, with 0.69 cellular metabolic processes, protein processing, and peptide metabolic processing. So all these results again can be obtained from this full results. So here it gives us full results for our protein in Excel. So here are all these results: fibrinolysis, regulation, response to external stimulus, and all these are open in Excel file. And if we look at our protein, so here is that protein which is similar to our protein. So here is our query protein, and here is the template which is shown here. So if we click only on this one, so this is query protein, and the other one is our template protein, and it gives us alignment or homology, that how much it is similar to our protein. Predicted enzyme commission numbers, top 5 enzyme homologs in PDB. So if we look at the enzyme action of this protein, it gives us 5 top enzymes which are homologous or which are similar to our protein, and also gives the enzyme commission number of that enzymes, like here 3.4.117.20, which is mostly aligned with RMSD of 2.30. TM score is 0.844, and similarly C score is 0.588, and it is in ranking in 1. And here we can click to view that protein. Similarly, predicted ligand binding sites, it also from here we can spin it on or off, like here it shows it is spinning. And predicted ligand binding sites, so template proteins with similar binding sites. So here we have different template proteins which have similar binding sites. So here we have 10 different proteins which have similar binding sites. If we look at here, this protein, what is this protein that binds with zinc? So it is zinc binding protein. So it is carboxypeptidase, complexed with 2-benzyl 3-iodopropionic acid. So this protein has similar active site according to the prediction of our Cofactor tool. Again, we can spin it on and off, and we can download all the predicted results from here. Results in, this is a compressed file, so from here we can obtain our results, and we can also give the reference of our predicted results by using these references when we cite our work in different articles or in our thesis. So, and then after viewing all these results, we can again go back to server, and we can again submit our next results. So here we have back to server, or from the left corner, top or bottom left corner, we can go back to the server. So this tool that we used and we discussed